It's my great honor to introduce today's seminar speaker, Professor Anderson Friedler. Professor Friedler is currently the James Eisner Chair in Chemistry, and he is also the Director of the School of Chemical Science in the University of Illinois. He received his PhD in Chemistry from the University of Arizona in 1988 and spent 3 years at Stanford before joining here. Professor Friedler's research theme involved both analytical chemistry as well as neurochemistry. He developed Acetylcholine versatile tool and also used this tool to study the distribution and dynamic release of neuropeptide and classical transmitters as well as their metabolism. Professor Friedler is one of the leading scientists in his own research area, and he has authored more than 340 manuscripts and have delivered more than 350 invited lectures. So acknowledging the impact of his research, Professor Friedler has been received numerous awards, including the Ralph Eden Award from the Pittsburgh Conference, the Field Award from the Eastern Analytical Symposium, and the 2014 ACS Award in Analytical Chemistry. Today, Professor Friedler is going to give a lecture about his recent work, and so please join me in welcoming him.
Thank you. Thank you all. It's fun to be here. It's always strange to realize that, I go around and, like most faculty, and give many talks, and then very few on our own campus about our research, lectures and other things. I'm an analytical chemist that studies the brain, and so I develop new tools to understand how the brain works. This is actually, we were just talking about this a minute ago, has become, I guess, a hot topic because this kind of fits in with Obama's brain initiative. Actually, every president over the last 20 years has picked one area, whether it's nanotechnology or brain or even the war on cancer, a little older than that, and Obama's has been understanding how the brain works. None of these initiatives have been successful if you mean that they've actually answered the questions they set out to while the president was still president. So I know Obama's will be the same way. We won't understand the brain anytime soon. It's actually kind of fun to be here. My first ever outside peer-reviewed grant that I ever got was a Whitaker grant from BioEngineering, and the chemistry department head said, what is Whitaker and what is BioEngineering? At that time, there was no department of BioEngineering. There wasn't much really going on here 20 years ago. There was some very important people doing work. And so I've always felt very warm feelings about bioengineering because, of course, that was my first, as I say, funding, which is, as a faculty, one of the important areas that we need to do. So what I want to do is kind of go over some of the tools. Now, from an engineering point of view, if you want to interfere, hopefully for alleviate, for example, disease in the brain, you have to understand how it's functioning in the first place. I usually end with this slide, and I decided to start with it because that way I always, I have time. The group has changed a little bit. This slide shows outside of the Beckman Institute a few years ago. It was on April Fool's Day, so the group was making fun of me and my hair. My group really, to join the group, you have to have hair that's bright colors, and no, it's so this is the group then. I became an editor of an ACS journal, administrator, running the director of the School of Chemical Sciences, and the group shrunk a little bit. It's probably in a growth phase, but these are the people that did the work. I have a lot of collaborators, some of which are listed here because I'm talking about their research. So let me give a few slides of background about the brain, and then I'm going to go into how we measure what's happening in individual cells in the brain. So, this is laser pointer's not too bright. This is obviously a picture of a human brain. We don't do anything in the human. So rats, mice, and other animals, I'll talk about those in a minute. This is a network of neurons, or a piece of, artist's rendition of the brain. And there's two things. One is brain cells are unusual shape. They have wires connecting them. The interesting thing is a microliter of mammalian brain has 100,000 neurons. It probably has a couple 100,000 red blood cells, a couple 100,000, 400,000 or 500,000 helper cells, astrocytes, oligodendrocytes, and others. And so if you take a microliter of brain and you analyze its chemistry, you actually really have not analyzed what's in neurons. You've analyzed all the helper cells. There's a lot more of them. But again, there's more red blood cells in your brain than there are neurons. So I show this slide. I had somebody saying, "Are you trying to say that my student's brain is mostly empty space?" I guess that's another alternative explanation. The other way to look at this is that even though the wiring in the brain is important, it's, you can't really change that for an adult too easily. And so it's the chemistry that is the mode of pharmaceutical intervention. The chemistry occurs in synapses. There's a picture of one. A synapse is truly nanoscale. You see in textbooks it's 30 to 50 nanometers or so. It depends on the synapse. Some are much larger. And so it's the chemistry that we can do something about. And then, to be honest, I never worked with drugs of abuse in my research. And the National Institutes on Drugs of Abuse, drug abuse contacted me because of the millions of compounds in nature, the 100 or so that affect this type of chemistry are known as drugs of abuse. And they hit the same types of circuits that the molecules in the brain normally do. One more basic slide again. Most people think of cells as small. I have cells in my body that are in my spine that go all the way up to my brain and down to my toe. They're not straight, so even though I'm not quite that tall, they could be 2 meters long. They're not part of the cell cycle. I'm more than 50 years old. My neurons in my spine are 50 years old. And so when people talk about cells as small and they talk about how they keep on changing, remember neurons don't follow any of those rules. And so you could even ask questions about how neurons take insult, chemical, physical problems that keep on accumulating during your lifetime. Whether or not a message goes from the cell soma, the cell body, down to a terminal depends on all these other connections in and around the cells. And so we want to understand. Okay. Yeah, that's a better one. Thank you. Now we have a laser pointer. So whether or not this signal continues depends on everything that's happening in and around there. And so we want to be able to measure that. Now, when I started this work 20 years ago, I kind of thought all the neurotransmitters, all the neuromodulators were known. I talked to lots of people in bioengineering. They look in a textbook and they say, oh, here's the growth factors. What I kind of want to do today is tell you, show you the techniques, but also tell you in every single system we've ever looked at, many neurotransmitters, neuromodulators seem to be not known, and in many cases they're quite a surprise. So everything about 20 years of work has really been dealing with complexity. Chemical complexity, spatial complexity, and temporal complexity. And how do you do that? One is obvious. I'm an analytical chemist. I like measurement tools. If you want to look at small molecules in the cell, you need a big tool. And it's actually, I think it's an unwritten rule, the smaller your sample, the larger the tool you need. And so if you want to look at really small cells, you need to have lots of money. I'm not really sure. I mean, that's true here for, fabrication. The smaller the line you want to make on a device, the larger the tool you need to make it. So we have some commercial mass spectrometers, and this is a time-of-flight instrument. This is one that Eric Lanny is just finishing at the Beckman Institute. It's started on a commercial instrument, and it's been home-built or laboratory-built for 4 or 5 years, and this is an ICR at the IGB. So we have mass spectrometers. These work great, and this is a large part of my research group, but I'm not going to go into how mass spectrometers work too much in this talk. This is a fun one that nobody really thinks about, and this has been hammered home in, actually as I look at science over the last 100 years. If you want to cure a disease, if you want to understand something, step back and think what you really need to know. When people wanted to understand electrical activity in humans, some people started working at mice and rats, other people did other animals. The group that was smart enough to go to the squid giant axon got a Nobel Prize because they could fill up something this long with seawater, change the sodium or potassium levels, squeeze it out and do it again. And it's a great model system. If somebody wants to do a genetic study on humans and want to do 10 generations, well, come back in what, 200 years? Well, of course, you use a mouse, then it's maybe a graduate career. Work on a fruit fly, which in some ways is a completely, I'll get people mad, irrelevant, lousy model in every way, except the generations are really quick. And you can manipulate it. And because of that, it's worth working on. And so biology, over the last 100 years, has had a number of successes with, in some ways, lousy models that answer really important questions. This one got the Nobel Prize with Eric Kandel for learning and memory. And I'm going to show you some data. This is some work with Rainer Gillette, and I'll just give you one example because it's lunchtime and you're not eating candy, you're listening to me. This is a Pluribranchia californica. It's a sea slug. It's under seawater, and it eats a Spanish shawl. Now this is bright purple and orange because it's poisonous. If you're a fish with eyes, you'd say stay away for something purple and orange. Unfortunately for this sea slug, this guy, or this hermaphroditic self-fertilizing sea snail, doesn't have eyes, tries to eat it, gets stung in the mouth, and spits it out. Its brain, which has maybe 1,000 neurons related to feeding, remembers never to eat one of these again. So they can learn. They don't learn bioengineering. They don't learn, English language. They learn how to find food, how to avoid being eaten, and how to find a mate. But this is a great model for learning. And then, I've done work with Gene Robinson, and plastic changes is you change the expression of a single gene in a bee, and you can go from a nurse to a forager, and then mice and rats. And this is just simply to remind people we're at the University of Illinois, and if you ever see a smiling cow, run away. We have actually done all of these models, and I will say here's a good example. This is why I like working with sea creatures. This is found off the coast of the state of Washington. You have to go there over the summer, collect the animals. You can't let anybody else do it because then you don't have an excuse to go to the beach. And this animal has two behaviors. It can swim and it can swim. Now, as a chemist, I can keep track of those. One is feeding swimming and one is escape swimming, and you can look at how that changes. And so every one of these animals has good reasons to study it, and they make the answers much clearer than if you work with something like this or even a human. And so these are really fun. The last way you can simplify is to go smaller. This is a honeybee brain. It's a whole brain, but it's a honeybee. This is about 1 million neurons. This is 1 million neurons from a brain slice from a rat. And I bet some of you work on neural nets and you do programming. Well, this is a real neuronal network. This is the cells that control reproduction in a sea snail, except we put them on a microscope slide, and I want to know what changes as this goes into fictive reproductive behavior. And so you want to know what chemicals are there, when they're there, and when they're released. And so chemical, spatial, and temporal information, and down to single mammalian cells. So we oftentimes work here and move back and forth using a zoo. In the last 5 years, I think we've had, now we're working on monkey brains and birds and planaria and snails, and actually we have a paper being reviewed in Nature on ctenophores. That's comb jellies, because it's the animal with the simplest nervous system that exists. Sponges are slightly simpler animals, but they don't have nervous systems. So we work at a zoo, and my group oftentimes thinks that they're the zoo when I refer to them that way, but it's the animals. So we work on a range of animals. We try to make and understand what's happening in single cells. Single cell measurements are hot. If you go to NIH, they have something called Directors Cross-Cutting Initiatives, or now, there's the Common Fund is another way to say it. So the director of NIH picks areas like, to be honest, last year, metabolomics and single cell measurements and all the areas that I work on. We talked about that a minute ago. That worries me because once you're working on an area that's really hot, it means in 2 years it won't be. But right now, single cell measurements are considered new. I think that's kind of funny. Leon Hoek, a few years ago, came up with the first microscope, and he published some notes in the Royal Society in London. And his notes basically identified things like red blood cells, single-celled organisms in pond scum, semen, lots of other cells. He just discovered an amazing number of cells. Cahal, analyzed neurons, and if you look at this, there are all sorts of different shapes. To be honest, these people are the ones that got to name the cells in our brain, and they got to name them using things like pyramidal cells or stellate or others. So they just named them on shape. And so people think that all the neurons of a similar shape are similar. They're not. If we do chemistry, we'd come up with a different naming system. About 50 years, people have been doing single-cell chemistry. This is a crazy example I love because it was so ridiculously ahead of its time. This group did single red blood cells. They published this paper in Science, and then every one of them quit and changed to another field because it was so hard. But they took a silk fiber. I'm actually not joking. They took a silk fiber in a humid environment. They put one red blood cell manually on one spot, put 1,000 volts across, and then the proteins would move across this silk fiber. They couldn't even buy capillaries, easily at that time. And then they would detect things using a weird interferometric optical approach. So what's changed in 50 years? Mass spectrometry, among others. So we do a lot with mass spectrometry. I will show you an example later on of mass spectrometry analyzing a type of brain chemical called the neuropeptide. In 1970, Guliman isolated a neuropeptide from a sheep, and it took them 4 million sheep hypothalami, the brains of 4 million sheep, to isolate enough material to analyze. Now you can do that in a single cell, so that's 15 orders of magnitude improvement in sensitivity. I always am amazed by that, but I'm also amazed that I can imagine the graduate student who works with the slaughterhouses, and they grab 4 million sheep hypothalami. They have the final thing going from vats down to barrels, down to a single flask, and you show your research director the flask containing all the biologically active peptide, and you're ready to do the final purification, and you drop it. You know, there you go. Oh, I guess I need another 4 million sheep. Anyway, things have gotten a lot better, and it's because of mass spectrometry. I'm going to not go into details too much, but this one allows you to do single, metabolomics, or the small molecule content, and these supply imaging. This one, SIM, secondary ion mass spectrometry, exists at MRL, so anybody can use it if they get trained on it. And all of these allow you to look at what's happening in neurons. And so ignoring this picture, I'll show you how well it works in the next few slides. So here's our sample. You can take a creature. This cell is green. Isn't that convenient? So we're going to isolate that green cell in a droplet and analyze it. By the way, some of the experiments we do are heroic. What I mean by heroic is they're much harder in this creature than in a mammal, because no one would do this. We've worked where at Columbia University, one of the groups that's actually worked on the gill withdrawal reflex, the part that got this the Nobel Prize, and they stick an electrode in, and they stimulate it, and they see if they get the right behavior. If they don't, they pull the electrode out and stick it in. Pull it out, stick it in. Pull it out, stick it in. Pull it out, stick it in. 8 to 10 hours, they find the cell of interest. So we're not doing single cell measurement like you take a cell off of your skin. We're using one cell in the entire creature. It has to be that cell. They then take this brain with the green cell, put it on dry ice, regular ice, so it's still liquid with seawater, send it to us, we isolate the cell, now it's not looking too green, and we analyze it. So these are hard experiments. The hard part was done somewhere else and here. So this is low throughput, hard experiments. I decided to do one example of not mass spectrometry, because I thought a lot of people who use optics. The type of separation we do is capillary electrophoresis. Imagine a hair. It's the same diameter, filled with seawater. You put 20, 30,000 volts across it, call it 20,000 volts. Charged molecules migrate. Positive go towards the negative side, negative go towards the positive. And if you stick a cell on this side, you get things falling out the end of the capillary. If the end of the capillary is a UV laser, instead of green, it's, say, 257, anything aromatic, you can detect. So I had a grad student build this instrument with Whitaker money. And we're still using aspects of this. Actually, I don't know how many people saw Warren Chan when he was here yesterday or earlier this week. So Warren is the one that actually really built this when he was an undergrad in my lab 20 years ago. And so Warren built this device. He thought it was amazing that it still was there. So he started out as an analytical chemist before, I guess, seeing the light, or whatever you want to call it. But anyway, this is Warren's and Aaron Timperman's device. So Warren got an undergraduate degree and Aaron got a PhD. I guess that's the way it works. And this was done a little bit later, but this is now when you have these sea slugs, wild caught off the coast of California. We pay a surfer dude. The business office hates this, but those are the only people that'll collect them. And so if the waves are up, they surf. If the waves aren't up, they catch these animals, ship them to us for money, and we collect, we get the animals. But these are pluribranchia. And we take one cell, the metacerebral cell, out. We analyze the serotonin levels. Now, you're going to say, what's serotonin? If you're in America and you look at the popular press, you've heard of SSRIs, or serotonin reuptake inhibitors, in depression, or monoamine oxidase, or MAOIs. Every one of these drugs are worth $1 billion plus, actually I think $10 billion a year, because they change your mood, and most people like to feel better. Serotonin in every animal exists, and it also has something to do with feeding. This is the first time anybody ever showed that serotonin levels in a cell can actually regulate an animal behavior state. Lots of variability, but these are wild-caught animals. I know some of you will probably work on rats and mice. These are even more wild. These are wild animals. We don't know anything about the history. The student who did this work did it blind, meaning Nathan Hatcher actually fed some of the animals. We catch them off the coast of California. They're shipped to us. We starve them for a month, and then we randomly feed one squid and one stays hungry, and we make these measurements. So this type of measurement can be made because we have an instrument that really only sees serotonin, HIA, and a few other compounds. Just to give you one example, in a mammal, if you take a cortex of a rat and you add serotonin, you see serotonin. You know it's serotonin because of its fluorescence properties. It is elutes at 7.4 minutes. But we also, HIA, a catabolite, the conversion of this to this is what some of the drugs deal with. But we actually found an unusual, unknown molecule that we now know is 5-hydroxyindothiazolidine carboxylic acid. I put this up because there's an example of a molecule that wasn't known to be in mammals. We've actually, and is it important? Well, every one of the catabolic processes of this molecule actually are the site of a lot of pharmaceutical intervention. So this may be important. The reason this works, though, is there's 10,000 compounds here, and we only can see those that have this indole ring. So I made a very selective instrument that only detects this part of the molecule. So that works if I'm studying serotonin. It doesn't work if I want to see everything else. By the way, those are big cells. Here's a fruit fly. This is just kind of showing you how good some of my students are at manipulations. I'm drinking coffee. Some of my students never touch coffee, and they have hands that are unbelievably fine. And so you can take a single neuron here. They're green again. You can pull one out. They're green because we've labeled them using molecular techniques. And you can actually now see serotonin and some catechols in a single fruit fly neuron. This is unpublished, but the difference between a 500 micron neuron and a 5 micron is 100-fold cubed, or 1,000,000-fold less material. My student takes this cell out and actually manually sticks it into a capillary the size of your a human hair. And so if you're good at manipulations, you can do some neat work. Now we'll go to mass spectrometry. So there's the green cell. What's in the cell? And so rather than pre-select for serotonin with a laser, like the laser-induced fluorescence system I just showed you, we're doing mass spectrometry. That gives you supposedly everything. I'm just giving you one example here, acetylcholine. Now, acetylcholine is a neurotransmitter that is a pain to detect because it's not fluorescent, it's not electroactive, it doesn't have a UV absorbance. I don't know how to analyze it. So I've spent 20 years, or actually about 18 years of my life ignoring acetylcholine because I can't detect it. And so the best thing you can do when you write grants and publish things, if you can't detect a molecule, you just pretend it doesn't exist. And that works, actually. We've gotten funding for molecules that we can detect. But we finally have mass spectrometry working in the last few years, and this is acetylcholine. I know it's acetylcholine because of its mass, and even though it's a single cell, I get a good signal-noise ratio, I get good resolution, everything works really well. Just to show you one other thing, we're measuring the mass of the molecule, and it's 146.12. Here's one single cell. It's a mass, or electropherogram, so things migrate out based on mass to charge. This is intensity. And we actually have 4 compounds in that cell that have a mass of 146.12. And only one of them is acetylcholine. So this is this weird technique called tandem MS. You basically take your molecule and you smash it on the ground and you look at the pieces, and only acetylcholine breaks into that piece. So we can do this for any molecule that's at high enough levels in a single cell. And every cell is different. I've had many people ask me this, okay, neurons are neurons. Well, these are these electropherograms with different colors representing different masses, and acetylcholine, serotonin, glutamate, betaine, we actually detect 200 to 400 compounds in every cell. I don't know how to show you that, so I just picked important molecules here. And every cell is a little different. One cell in the entire creature, the R15, has this mass. I've never been able to figure out what it is because it's only found in one cell and there's not enough to identify it. But it's probably important. Every one of these molecules, by the way, are found in your brain. So I guess I haven't really emphasized this so much, but every animal we've looked at, when you go from a coral and a jellyfish to a human, the neurotransmitters are the same. And that makes what I'm doing possible. And so, who knows about that one, but most of the others are really the same. And you can get data that looks like this. And I actually am showing you this slide because my students usually misinterpret it. But this is the metacerebral cell. It's the only cell that has serotonin. And my students will come to me and say, look at all the biological variability. Each animal has a different amount. And I like this slide because it turns out that's not true. What I already showed you is this variability tells you how hungry the animal is. It's not really random. And so the the important point is when you measure something and you get a lot of scatter, maybe it really is variability, but maybe what it is you're not controlling the right variable. We've actually had other cases where we see a huge amount of variability, and then I discover if we always sample at 8 in the morning when the lights come on at 6, then the variability goes away. And so biological variability may be just that, or it may be something you haven't controlled. Yeah?
Is there some connection to circadian rhythm?
Actually, with serotonin, I didn't show it. Serotonin and serotonin sulfate does follow circadian rhythm. We actually did a paper on that. That was actually because I couldn't understand serotonin sulfate. I had a graduate student making measurements that was free running. Sometimes would make measurements at 3 in the morning or 5 in the morning or 7 in the morning or 1 in the afternoon. And after seeing values that went from non-detectable to micromolar, after doing this 50 times, he did something that was quite important. He looked at his notebook very carefully and noticed that luckily he recorded when he took every sample. And he noticed that this followed a circadian rhythm. We didn't think of that when we started, because it's not melatonin, it was serotonin sulfate. So yes, there is a circadian rhythm. In our body, there is a seasonal rhythm between melatonin, which is made from serotonin, N-acetyl to melatonin and others. And so that is and there's a seasonal variation. Let me show you one example, because I know people who work with cell lines tend not to like to hear this. There's 2 neurons in aplesia. It's the last example I'm going to give you from the sea slug, I think, that are called B1 and B2. There's something to do with buccal and feeding, so they're 1 and 2. And we isolated them from an animal, and we analyzed them as fast as possible, or we cultured them for 1 day. So all of these cells are freshly isolated, 1 cultured per day and 1 not. Here are the B1 cells and the Cs, meaning they're cultured. And the chemistry changes. Now this is a weird plot. It's principal component analysis, looking at all 300 to 400 peaks that we detect. Actually, in this case, I think it was just the 50 most intense peaks between these, and these are separating, meaning there's something different about the chemistry, the levels between the B1 cells and the cultured B1, but for the B2 cells and the cultured B2, they're not different. And the reason I'm showing you this is this slide. B1 cells and B2 cells are distinct. They do something different in the animal. And here they are, the B1 and the B2 separate on this principal component plot, but after 1 day in culture, whatever made them chemically distinct is completely gone. So this is a cell line. This isn't a cell line. These are freshly isolated cells that were only cultured, primary culture, for 1 day, and whatever made them different is gone. And let me tell you, we're good at culturing these cells, and we can't keep them. So whatever happened in the intact animal disappears. So as you're doing experiments in microfluidic devices, and we do that all the time, and others, be aware there's nothing less physiological than culturing a neuron in a device. And chemistry changes very quickly. And I don't know what the answer to this is, but to also sometimes work with intact cells if you can get away with it. Things change quickly. Here's the hardest experiment we're doing. And it's the names, completely ignore the names, but we're working with 2 brain regions. This was with Lee Cox, and we're still doing it. The ventral basolateral nucleus and the thalamic reticular nucleus. Okay, that's a mouthful and doesn't matter. We're interested in why we see GABA where we shouldn't. And so this is the enzyme to make GABA. I'm showing you this, not for this data, but to say we went to a rat, and in the 2 areas we can do this mass spectrometry, and in this area we see acetylcholine, GABA, and glutamate, 3 important neurotransmitters, and in the VB nucleus we see others. Okay, nothing new about that, but I wanted to take this to single cells. We decided to try and see if we can know what cell we're sampling. So we're using something called patch clamp method. Anybody here heard of that? No, good. So this is patching onto a cell. Actually, Nair and Sackman did this in, I think, 1991, got the Nobel Prize for it. Actually, when you do this, you have access to the cytoplasm, and we can suck up a little bit. So here's the physiology of the cell in the brain slice. Here's another cell. Some of them are spontaneous active, some are not. But we can suck up. This is the original cell, and the movie is so hard to see, and this is hard to see, but it's 9.6 by 8.5 microns. And after we suck up about 3 picoliters, it's 8 by 7.3. The cell is alive the whole time. If we really do things right, we can actually get metabolites from those cells. So here is the physiology from spontaneously active cells, from TRN neurons, VB neurons. This is an astrocyte. Here is the shape because we injected a dye in, and these are just partial metabolomics traces showing you GABA and glycine and serine and other compounds. So this is the smallest sample we've ever done. It's the first time I've ever seen anybody do something where you actually take a living cell, you can actually do physiology, morphology, and chemistry at once. And so this gives you a lot of ability. For example, NIH has all these calls for proposals because no one knows how different astrocytes are from each other. Or, people like to say astrocytes are astrocytes. I don't show you more data because we've now done about 10 astrocytes, and the chemistry in different astrocytes is amazingly different. We see different compounds, and we haven't done enough to understand what we're seeing. So this is known because of this part of it, but the chemistry that's occurring in astrocytes is pretty, very poorly known. Okay, that's one type of mass spectrometry, and everything I showed you is what's happening in cells, but isolating the cell is hard. So now we're going to go to imaging. Now, I bet a lot of people here use microscopy of sorts, fluorescence microscopy. You can actually do mass spec imaging. It's horrible resolution. It's lousy imaging. What you do is here's a brain slice, and here are a bunch of red dots. You can shine the laser there, or shine an ion beam there. Things will explode off, and you can do mass spectrometry on it. I move it over 20 microns or 20 microns. If I do 1,000 by 1,000 array, I get 1,000,000 pixels. Each one at 10 micron or 20 micron resolution. Nobody in the right mind would do this, except at each pixel I get an entire mass spectrum. So it's lousy spatial resolution. It's hard to do, but you get an amazing amount of chemical information. And so we can use a laser. See, I'm using one up here, and I get about a micron, really usually 5 microns or so. I can do proteins sometimes. I can use a gold cluster. That's certainly for others. We're using buckyballs up at the Beckman Institute, and you get a smaller area, but it's actually smaller molecules. And you can take a single cell. There's a cell, and you can actually see lipids. You can see vitamin E localization, and this works quite well. So you can image cells and their lipids, for example. You can even see what happens as you go towards a simple neural net, and you see what changes as, so this is these 3 cells, here's one of them, and here's just a terminal, and we get some information on phospholipids. And so this is some work that we're really just, ongoing and trying to make work. You can combine these. This is actually some, we're still continuing with work like this. You take a section of the spine, you do SIMS for lipids, you take the next section and you do MALDI for peptides. These are peptides here that control behaviors. You take the next 6 sections and you do chemical analysis, and you can work your way down the spine. And we've done this and found lots of different things. This is the first images from a new instrument we just built. As I say, it took about 5 years to build. And the reason this is good is because it gives us more information about some of the lipids. So it's this buckyball SIMS imaging. It's on the 2nd floor. And you can actually get information on higher quality mass spectra than what I was showing you. So here's 2 cells. The imaging resolution is not very good, but the mass spectral data is quite impressive. There's lots and lots of lipids in that area. Just to give you a feeling for this, by the way, Lipid Maps, a consortium of groups that NIH has funded. Basically, you hear that there's 20,000 genes in the human genome. There are 600 to 800,000 molecular entities in your human brain that are called lipids. Right? So different length chains, different sites of unsaturation, different head groups. So, and the other thing to think about it, and everybody gets surprised when I say that, is if you wanted to go into your refrigerator and find the item that most resembles your brain, and you had a chicken breast in there, for example, most people say, oh, that's a good model. No, to be honest, your brain is like a bar of butter. A little bit of protein, a little bit of water, but it's about 70% lipid. It's oil, it's fat. And those lipids turn over, and a lot of function of the brain depends on them. And there's no easy way to analyze them. So the reason these types of tools you're starting to see a lot is because no one really even understands how lipids in the brain turn over or how they work. And so I will say, when I tell some people that, they get very upset because they don't like to have their favorite, most prized possession, their brain, be compared to a bar of butter. I came up with that because I'm an editor of a paper, and a reviewer and an author were going back and forth, and somebody had actually used chicken breast as their background approach for their mass spec imaging, and the reviewer said you shouldn't be using a chicken breast, you should use a bar of butter, and the reviewer got really, really mad, saying, how could, you should throw this reviewer out because he's comparing my brain to butter. So I looked it up, and the reviewer was right. Anyway, so let me end with a little bit about neuropeptides. Neuropeptides are the hardest molecules to analyze because they're present and active at really low levels. I already mentioned they used to take a lot of work to isolate. Some books, some classical textbooks say that 4 of the 7 deadly sins are mediated by neuropeptides. Gluttony, attraction, and things like that. So they're important, or not. Mass spectrometry has gotten really kind of fun. You can take a section of brain, you can isolate a cell, or here's a neuron. If I put TNT, the explosive, around the cell, and I shine a laser here and it explodes, I get a spectra right at that spot. And that spectra, every one of these peaks are the peptides that were in that cell. It really works well. This is sensitivity. This is a chemical printer printing spots of the peptide angiotensin on here, starting out at 60 femtomoles a spot down to 3, keep on going down to 300 zeptomoles per spot, and we still can detect it. Our detection limits are about 100 zeptomoles. So people say, what's a zeptomole? It's 10 to the minus 21 moles. That's really good, except the zeptomole is also 600 molecules, so this is about 100,000 molecules or so. So it's good, it could be better, but it actually allows us to see what's in a cell in terms of peptides. This is older data, but we're the first group that ever did things like take a single vesicle in a single neuron, in this case a big one from aplesia, and we can see some of the peptides are present in there. Some vesicles that look different don't have peptides. You can look at a cell in a process, and everywhere we stick this explosive matrix, we get peptides. And so you can see lipids and peptides going from a cell soma down a process. And this kind of highlights that type of information. Here's a bunch of cells that we labeled with an in situ probe. You can pop a cell out and do mass spectrometry on it. And that cell is missing because we actually analyzed it and we got the neuropeptides that are present. We've done this probably 40 times and discovered for publications like the Journal of Neuroscience, the neuroscientists don't even know that single-cell mass spectrometry is weird or difficult. They just come to accept the fact you can do physiology and mass spectrometry. If I know that this cell makes this animal go into swimming, when I do this and I discover a new molecule, I, even as a chemist, can say maybe this molecule has something to do with swimming. And then we test it. So we've discovered literally thousands of brain peptides, but we actually have about 100 that we know are neuropeptides. We have biological activity. And just to kind of end this type of thing, you can go even smaller. This is a dorsal root ganglia, those cells in my spine. As I say, I like these because they're such strange neurons. They're long. They're probably the largest animal cells. Let's see. Oh, I didn't. Sometimes I show a picture of a blue whale because a DRG neuron could be, in a blue whale would be 100 feet long. That's a single cell. And so we can look at those in many ways. Here we've taken a single process from a single cell, and each one has different chemical profiles. And so this one has one peptide, this has a bunch of unknown peptides. And so we can make this work really small. I don't know any other group that's doing anything like that. We can use, in a mouse, molecular approaches. This is a green cell. We're good at isolating green cells. And every one of those, we see the peptide profile of different green cells. And it says here, how do those change with pain? You're probably thinking that about now as towards my end of my talk, is when is pain going to end? But we can take a dorsal root ganglia group of neurons, separate it in a microscope slide. There's some cells right there, pieces of fibers. You can separate the fibers out if you want. But we can image this with mass spec imaging. And this is 1,000 individual cell spectra. This is 300 that were undergoing a painful stimulation. We injected something painful into the foot of a rat. 300 on the other side, water injection and control. And 4 neurons, this is a mass spectra from 340 to 348 Daltons. 4 neurons had a huge change. Went from, here. But actually the other 300 or so didn't change. So we can find the rare cell using this approach. And this is the same data shown differently using 3-axis principal component 1, 2, and 3. Here's 1,200 or so cells. And we're interested in the outliers up here because they have different chemistry. This is different because we're looking at one mass, in this case of one of the peptides, at 1100.8. And we can actually take that cell, one of these cells of interest, and analyze it again because most of it's still on the target. And so we can take that weird cell after we've done mass spectrometry, 90% of the material is still there, and we can then do CEMS on it. So this is capillary electrophoresis mass spec of the metabolites after the last data. Now, let me show you, I have two students trying to make this better. One actually is mostly working on software. This is these red dots are the, these black dots are the cells, and this is rastering the laser in a normal beam, and you obviously, wherever there's a red dot, you got a little bit of the cell. But what we're doing now, instead of this approach, is we actually image the locations of the cell and only shine the laser at the cells. So we're not doing imaging anymore, we're actually just finding the cells. So this took us a while to make work. This is a really recent data set, but we imaged using dark field and autofluorescence. We found the cells, and then we did a 3x3 imaging, raster around it, and we hit the cells really well. And wherever we hit the cells, we get a mass spectra. And this is the most recent data. This is 2,000 cells. We did a principal component analysis on it. So here's the target with lots of cells all over. This is principal component analysis. And these weird, most of the cells are here, but some of them have weird chemistry. And I'm just showing you random spectra, of a few cells around that spot, in different locations from this principal component analysis. So if you don't like the brain and you've forced yourself to sit through awake this entire time, imagine looking through, for example, using cancer. And you wanted to find the rare cells that maybe first started to change. Doesn't matter what it is, whether it's cancer, whether it's developmental and you're looking at stem cells, or the brain. This is a way of probing with chemistry large data sets and then trying to extract information. The problem really is that it takes us actually a surprisingly short time to get this information, and we're just learning how to go through 2,000 cells, each one with a gigabyte of data in terms of the mass spectra, gives you terabytes of data in the wrong format to do this. And so we're still learning how to make that work. I want to conclude, except I have one more example after this, but just in terms of single cells, are we actually done? Have we done enough? And the real interesting thing is that, from a proteomics point of view, you can't do single cell proteomics. RNAs, you can, because of Eberwein and others, you can do single-cell transcriptomics, and we're really just starting with metabolites and lipids. But if you look at a cell like this, it's not even the single cell here. We want to understand the differential distribution throughout this creature. Now, how much time do I have? Do I have 3 minutes, 5 minutes? What? What? Okay, perfect. So I'm going to show you, I'm running out of time. I'll give this example. Some work with Martha Gillette, just to give you more of a functional context. From every animal around, time controls everything. So if you think of it, it's obviously eating, sleeping, waking, drinking, heart attacks. To be honest, the National Institute on Drug Abuse thinks it's important that people abuse drugs more at night. I always think that's really kind of weird because, there's more crime at night, probably because it's dark, but they're convinced it's circadian, and so we're studying that also. So how does this work? With Martha Gillette, we're working and studying the signaling of the suprachiasmatic nucleus. This is a small 10,000 neuron organ in the brain. It controls all the body's clocks. If you take a rat, now I'm showing you a picture of a human, but we didn't do a lesion study in a human, but if you take a rat, if you remove the SCN in a rat, the animal loses circadian rhythm. Even 20 years ago, it was known if you took another SCN and stuck it close to there and just let it free float, you would restore circadian rhythm. That implies there's something being secreted by the SCN that can entrain all the body's clocks. In 2005, Science magazine published the 100 unanswered questions, top 100 answered questions in science. Now they can't count because there's 125 answers or problems, and about, I would say 100 of them were really good. What is the origin of the universe? What is consciousness? They're great questions. I just don't even know how to study them. But one of them was, how does the SCN control all of the body's clocks? And as an analytical chemist, I said, I can do that one. And so we started looking at that. So I'm going to show you progress in about 8 years with this type of, in this area. So the first thing we did is we took some SCN punches and we measured the peptides in it. And I hate long lists. But we found 102 peptides that were present there, including 29 that people didn't know about. Some were phosphorylated. So this is a smaller list. But it's too high. I don't know how to deal with 100, so we have to simplify it. So this is one of the approaches to do. This is a living brain slice. You take a brain, you take a slice out that has the SCN. There's little white dots because we put little collection beads that can grab onto peptides that are being secreted. We then wash them off and we measure, pretending like it's a cell, we measure what was in that bead. And what when we do that, we can see different things released at different places. This is arginine vasopressin, for example, near the SCN or the SON, and not in between. I'm showing you this because of this type of data. This is during the day or night. Arginine vasopressin is released at this time of day, and this mass, 1495.75 at night. There's no known peptide that does it, so that maybe is important. So we're getting some information that's kind of neat. You can figure out what that mass is. I told you, 1495, using all this complicated tandem MS. We fragmented it, and this is just a way of saying that with a ridiculously good E-value of about 10 to the minus, I think it's 10 to the minus 7 or 10 to the minus 11, we're able to show that this is cerebellin, a name of a peptide that was known to be in humans and rats and mice, but has no known function. So we can start to get some information. We can modify how we collect. Here's the brain slice. Here's the optic nerve. We can stimulate that as if this brain was seeing light. We can collect the peptides that are released, and we get data that looks like this. Before stimulation, we get a little bit of garbage and lots of background. We stimulate the optic nerve, the retinal hypothalamic tract, at 10 hertz at that time of day, and we get all of these peaks. Each one is a peptide. Then my graduate student and a postdoc spent a year. I'm not going to tell you about that, but that's really how long it took. And we identified every one of those peaks. That's not very straightforward, but we now can tell you when this says, for example, that it's big Len or neurotensin or PANC or substance P, we did a lot of work to identify them. That's not easy. So the real answer is that the SCN synchronizes the body's clocks with lots and lots of different peptides. It's not very obvious that it's one compound. You can even look at it this way. This is at one time of day, another time of day. These two are at the same. And as you change the frequency of stimulation, different levels of light coming in, you get different profiles. And so it is a complex system, even though it's only 10,000 neurons. We have made progress. There's a peptide there that starts with serine, alanine, alanine, serine, and it's a smaller form. And so somebody called it little stace. That peptide is secreted by the SCN, so we made the peptide and dumped it on. And this is showing you the phase shift of the brain as it sits there. And you can measure this. Martha's group is really good at that. These are Martha Gillette's group. These are electrophysiology or single unit recordings from the control. You add AVP, there's no change. You add little SACE, and there's a phase delay. So you've changed the phase of the brain in the dish. Turns out it doesn't matter if you add 100 picomolar or 1 micromolar, it does this phase shift. And by itself, stimulating the retinal hypothalamic tract does the same thing. So we discovered a peptide that seems to do at least part of the phase shifting. After we did this, somebody else made a little SACE knockout mouse, not us, somebody in New York, and they found that at this time of day, when you expose the animal to light, you don't get a reset of biological clock. And so this all does fit. And if you like to see more physiological data, this is some Gillette's group with Norman Atkins. 50% of the little SACE neurons actually respond to CFOS when you give light, and so that means they are light sensitive. So all of this fits. We discovered a neuropeptide in the circadian system using mass spectrometry, and now we know what it does. And then the true last example, VIP, vasointestinal polypeptide. This one we couldn't detect. We actually sent our original paper to Science, and 2 of the reviewers said if you can't detect VIP, the most common and well-studied SCN circadian peptide in existence, your system doesn't work. So we refuse to let go of that, and though this is Jennifer and Martha's group has done some functional testing, but we can't detect the VIP, but we detect a small form. And it turns out, after a lot of work, I can say our mass spectrometry was right, and the 500 papers that actually detected the long form were wrong. They actually just used immunohistochemistry. So this bar from here to here is the amino acid sequence of VIP. We only detect this. It's cleaved at an RK. And it turns out no one else has actually done mass spec on the SCN like this. But here is what happens when you stimulate that and you look. You see the small form of VIP is secreted. And if you actually wanted to know what it's doing, the same single unit recording, and VIP and the short form of VIP cause a phase advance. And it's a phase advance or delay, depending on the time of day. And so they both work exactly the same, and they both go through the VIP receptor 2, VPAC2. The only reason, if they're identical, why do I say this is biologically active? Because this is not released. This was isolated in the gut of a mammal. This is what's in the SCN. So now we can at least explain our observations and have information that people have been testing the wrong peptide. Now I am done, and I wanted to leave with 3 points. The mapping of the brain initiative of Obama, it certainly is a grand challenge. A lot needs to be done. The reason it's hard is really the dynamic range of the brain. If you think of distances, this is nanoscale, but in my spine I have things separated by meters. In the brain itself, by centimeters. The time period is milliseconds for some neurotransmission, and yet my neurons are not functioning as well as yours because they're 3 decades older, and it's the onslaught and oxidation and protein changes that occur during decades that actually changes things also. And the molecules range from nitric oxides to proteoglycans. And so in every case, there's 6 orders of magnitude dynamic range in length, time, and chemistry. And the other thing to end with is I have a lot of people who tend to think that everything's known in terms of chemistry. Right? And so I know lots of people coming up with a new tool. They said, I looked in a book, I found the 18 molecules of the cell that are of interest, and I'm going to measure those. And I assure you that when we use different tools to look, every cell we look at has surprising neurochemistry. I didn't go into it, but in some cells, the single most intense by mass peak that we see in the cells, we can't identify yet. So that tells you there's a lot of unusual neurochemistry still to be discovered. And that's true in mammals as well as some of the stranger creatures we work with. And truly, the last slide, I've had a lot of fun for 20 years, basically working with, a range of animals, and, even the Illinois animals like the cow, pig, and the honeybee, which I guess are more agricultural in nature, but have been also a lot of fun to work with. And thank you very much for your attention.
I wonder if you could talk a little bit about dynamics, thinking about brain activity and functioning. You're seeing the pain neuron is a good example. You're seeing activity, but it's not dynamic in some respects.
It's not dynamic. So I have, if you think about it, I kind of tend to think there's dynamic, there's chemical, and there's spatial. I know most people in this room probably wouldn't have put chemical as an axis. And mass spectrometry and the techniques we use give us a lot of chemical information at the cost of dynamic. So dynamic information, time resolved, is critical. I just have given that up because I don't know how to get it at the same time. Now with fluorescence, for example, you can make a probe and you can pre-select the molecule of interest. It could be a neuropeptide, it could be a small molecule, and then you can get dynamic and space. Now in one sense, you don't get chemistry except that you've built it into your probe. So if your probe only images nitric oxide, if your probe images serotonin, then by definition, you're selective. But I would say that that's because you knew in advance and you built it in. But you're right, we've given up a critical access of information to keep the chemistry. And we pay for that a lot and so I've picked problems that are slow. Time of day is great. It's dynamic. I've had people say, well, that's unfair. You're using something that takes hours. And I kind of think in my mind, that's good. I picked a good problem. If I can do hours, I'm going to work on time of day or seasonal changes. I can't do milliseconds. I've had a number of people say, well, you cheated. You picked aplesia with 300 micron cells, and they actually act like this is a negative. And I say, yes, I cheated. I picked a big enough cell to work with. Why would you pick a 3 micron cell that you can't get the information you want? And so, in one sense, if you really understand the brain, and you tell me that you have, and this is actually an important point, I spent 20 years learning this. If you want dynamic information and you have a particular molecule, there may be a part of the brain that's perfectly well suited. I didn't go into it, but we work with D-amino acids, D-serine or D-aspartate as opposed to the L-amino acids, and those are actually, like D-serine is glia-specific. It's only found in astrocytes, unless you move to the next region of the brain over, and then it's found in neurons, and in another part it switches back and forth, and so you get to choose. It's kind of fun, and so by knowing enough, you can actually decide that if you have a set of tools or criteria, you can actually pick the problems that are relevant, and in our case, we've given up, unfortunately, the fast.
So have you done anything on cancer yet, or this whole heterogeneity problem? You kind of alluded to it, but I'm just curious.
You know, it took, so we do a, what do I want to say? We don't do much or anything really with cancer, with the exception that we have worked with some cell lines, like immortalized cell lines, like ATT20 cell lines, or endocrine cell lines because they release, and they are a cancer cell line. I don't tend to think of them as cancer in the sense, I think of them as a model of an endocrine cell line. It's taken me a decade to learn enough neuroscience to where I actually can make my way around, and after 20 years I actually feel confident about some neurochemistry. When I think of cancer, it's, you know, it's going to take me another decade before I know enough to actually ask the right questions. And so, no, I haven't done anything really with cancer. Oh, it is. And so I've had a, you know, Amanda Humman at Notre Dame is a former student of mine who was using some aspects of this, and she's actually both in chemistry and in her cancer medical school. And there's other people doing this with cancer. And that would be great to look at, you know, how the environment in and around stem cells, because presumably that's partially chemical in nature, that helps determine the fate of a particular cell. And so I would think that these approaches are going to be very important when cell heterogeneity is important, and that's in certain diseases like cancer, it's in development, and it's in the brain. Now there's probably others, but those are obvious.
Do you find any differences in these chemical profiles between sensory versus motor neurons? I mean, there's clearly difference between astrocytes and neurons, but...
Oh yeah. So in aplesia, every cell is slightly different. The motor neurons tend to be big, and they have very big differences. So the peptide complement is different. So we find, you know, so we don't know enough in the mammal because we haven't done enough single cell. We've done about 40 to 50 using the patch clamp approach. We see, for example, in the TRN, I mean, this is a good example, these one set of homogeneous neurons. We measure, now when you do a patch clamp experiment, sometimes you don't patch on. We throw those cells out. Sometimes we think we've done everything right and we don't see the amino acids that are required for protein synthesis. We throw those samples out. But in 25 cases, we got really high quality data that makes us think we patched onto a single cell. And these are all glutamate using cells, and in 21 of the 25 we see high levels of glutamate, and 4 of the 25 we don't see glutamate. Our physiology friend says you're absolutely wrong, that can't be true. Everything else is right, the physiology looks good. Why is glutamate missing in 4 out of the 25? I don't know. We have seen now 4 spontaneously active TRN cells where you don't inject current and they're still active. And all 4 had high levels of histamine. I don't know why. So we're learning that. But the heterogeneity, you know, in one sense you should do 10,000 cells. And when we want to know the physiology within the context of the brain slice, then we're using patch clamp and everything else, and those are kind of slow measurements. And so we're still learning, but so there's huge differences within what's supposedly a defined population, and you change populations of cells, the chemistry is completely different. And I say that they still have all the amino acids, you're still doing protein synthesis. So when I say the chemistry is different, I maybe exaggerating. Transmitters, some housekeeping, a good example of a difference is in one part of your brain, you use nitric oxide. That's a neurotransmitter. It turns out 1 in 1,000 cells use NO. NO is degraded into nitrite. It's well known and established that your brain has about 10 micromolar nitrite. What we find is cells that use NO have 10 millimolar. Most of the people doing modeling of NO and nitrite say we're crazy, it can't be 10 millimolar, but that's what we get. However, if it's 1 in 1,000 cells, that explains their number. It's 10 millimolar in the NO producing cell, almost 0 in the rest, that averages out to 10 micromolar. Now, from a modeling of chemistry and how the enzyme works, there's a huge difference between 10 millimolar and 0. So that's an example where the heterogeneity is striking, and you really have to do it single cell at a time because you get an average otherwise, 10 micromolar as opposed to really high and 0. And we're still trying to understand that. The problem in that case, if it's 1 in 1,000 and there's no easy way to get to it, that means you have to do thousands of cells to get 10. That's kind of like the cancer. And so one of the reasons we haven't done something like cancer is I can't do 1 million cells to find the one.
More questions?